Okay, everybody, hope you're doing well. Today, in this lesson, we're going to go over passive transport, which is a form of diffusion. It's important that we understand how diffusion works because you have a lot of different substances that are capable of moving into and out of the cell. And these are generally substances that the cell very much needs. So this is going to be types of transport. This is going to be a two-part lesson. And the first part here is going to be passive transport. That's what this video and this lesson is going to be all about. All right, well, quick little prelude is one of my hobbies is I like to go out and paddleboard. I have a big paddleboard. I go out into the Garrison Channel. I go by Davis Island, Harbor Island, the Hillsborough River, sometimes out into Old Tampa Bay off Bayshore. And sometimes I'll just go out there and unwind from a day, relax, and I'll just sit on the paddleboard and really not do anything. I'll just let the tide take me wherever it goes, hopefully not into the wall. But sometimes I'll be in the Garrison Channel over by, let's say, Jackson's and American Social and Tampa General Hospital. And before I know it, I'm all the way down by Sparkman Wharf. And I didn't do a thing because the current is just taking me from one place to another. No energy, no motor, no paddles, no kicking, no nothing. Just going with the flow. And that's what passive transport is really like. Passive transport is the movement of molecules and those molecules can be of any type that does not require energy. And that energy is typically going to be in the form of adenosine triphosphate, ATP, the stuff that the mitochondria and the chloroplast produce. Now, in order for this to actually proceed and go forward, the particle, the molecule, whatever we're talking about, must move down a concentration gradient. From an area of high concentration to an area of low concentration. All right. Now, what the heck does that mean? Okay, well, let me describe a few things. Let me give you an example. And if I was actually in class with you guys during this particular lesson, we would actually do this example in class. So check this out. Let's say that this is our classroom. And what I would do is I would ask one of the guys and one of the girls if they have some sort of body spray or perfume that's aerosolized. You can spray it. And we've always got at least someone that has some sort of perfume. So let's say that you have this little sort of perfume or body spray or whatever. And what I would do is I would walk over here in front of the projector screen, like right here, and I would just spray it all over the place. I mean, back and forth, back and forth, back and forth, back and forth. And what I would do is I would ask you guys to raise your hand when you smell it. And so you know how my room is set up. We got all these students. Oops, I should probably use a different color. We have all these students. Each pink circle represents a student. Okay, so right now, this area is a high concentration of this this perfume or whatever it is I spray. We'll just call it a perfume. It's a high concentration. Everywhere else in the room, here and here and here and anywhere that there isn't the perfume is going to be low concentration. And according to the laws of just passive transport, these particles are going to spread across the room without any energy. You don't need a fan. You don't need to blow it around. It's just going to do it all on its own to try to reach a state of equilibrium. So I would say within 2 minutes tops, 2 minutes tops. This particle would be all over the room, and it would basically be equalized. And what would happen is, as we start to progress this way, more students will start to raise their hand, and then the students in the back row finally raise their hand, and we know it's gotten throughout the whole room. So generally, molecules are going to diffuse from a high concentration to a low concentration under the laws and the rules of passive transport. That is simple diffusion at its finest. To give you another example, imagine that we have a permeable membrane. This black line represents a permeable membrane. Permeable means that molecules and particles can pass through it. And imagine that I just have a lot of particles over here. And we're going to call this, let's say we're going to say this is 5 molar. If you haven't taken chemistry yet, or if you're taking it now, you likely have not gotten to molarity. Molarity is a measurement of concentration, how much of something there is within a given space. To give you a really crude example of this, or if we were to talk about, like, say, the hallways of Plant High School during class change, we would say the molarity of students is very high. There's a lot of people in a very small space. As opposed to when the hallways during class change, there's hardly any people in there, okay? So this is on the left side. We're going to call this a very high concentration, but high is subjective. You know, high compared to what? You can say that's high, but what if I have double the amount on the right? So over here, I'm just going to draw a few. This is definitely low in comparison. So according to the laws of passive transport, these molecules are going to diffuse across the membrane from left to right, which is from high concentration to low concentration, until you've reached a state of equilibrium. And almost forgot, let's say that this side is 0.5 molar, so just half moles per liter. So that's generally passive transport. You see this if you put food coloring in like a bathtub. Eventually you put several drops of food coloring, or you put the food coloring tablet into a big body, not a big body of water, but something like a bathtub or a toilet. And eventually the entire area is going to be that food color, because it's going from high concentration to low concentration, okay? So there are 3 types of passive transport that you will need to know. Number 1, simple diffusion. Now, warning, warning, warning. This is the form of passive transport that students forget the most. I think by now you guys are starting to know that I don't just make up these warnings. I don't make up these ahas. And you might be saying, oh, you keep saying this, it's not going to happen to me. Why not? I hope it doesn't happen to you, but so did the students that said that. So trust me, simple diffusion seems to be the one that students forget the most. And I have my hypothesis as to why. It doesn't have some fancy scientific name. It doesn't say osmosis. It doesn't say endoplasmic. It doesn't say photosynthesis. It's just simple diffusion. Not a very tough word. And I think it's so easy, you just seem to overlook it. So what happens during simple diffusion? Well, molecules move from an area of high concentration to low concentration through a permeable membrane. Now, I'm going to put something parenthetically, that means in parentheses, no pumps, no channels. All right? Just straight through the membrane. Now, we need to be very clear about something here. this is going to stop when equilibrium is reached, which generally means it's even on both sides. Okay? Now, generally, guys, in biology, more often than not, but not all the time, the membrane that is in question here is going to be a phospholipid bilayer, like the ones we learned a week or so ago. The same stuff that makes up the endomembrane system organelles, the mitochondria, the chloroplast, the nucleus, and the plasma membrane of cells. Okay? Now, a few things that you need to remember about this plasma membrane. It is nonpolar. The interior is made up of lipids. Lipids are nonpolar molecules, so only nonpolar molecules can pass through. And if you're still having trouble determining if something is nonpolar or not, if it is polar, it has a charge. In chemistry, polarity means positivity and negativity. If something is positively charged, negatively charged, or has areas of the molecule that has both, it is considered to be polar. It possesses poles. Nonpolar doesn't have that. Now, not only do nonpolar molecules pass through, but also small molecules. I'm going to make sure I mention that. You can't have something really big in nonpolar. It's got to be small. So with this in mind, let me ask you this. Could H2O pass through this? Just diffuse to the other side over here? The answer is no, because H2O is polar. It cannot pass through the interior of the membrane because the interior of the membrane is nonpolar. So let's get rid of that. So what kinds of things can pass through? Only nonpolar substances, and generally substances that are very small, okay? And I'll illustrate this all out for you in a little bit. But only nonpolar substances and small substances. All right, number 2, we have facilitated diffusion. Remember, this is all passive transport. The word facilitate means to aid or to assist or to help. Like I am facilitating your education when it comes to college level biology. I'm not doing it for you, I'm just helping you. Well, in facilitated diffusion, this is a means of passive transport, and when you hear passive transport, molecules are moving from an area of high concentration to low concentration. So that's something we need to make sure. That is a staple of passive transport. Molecules move from an area of high concentration to low concentration. Okay? Now, this is going to be within, or let me rephrase that. But with the aid of a channel protein. Now, why do you need a channel protein? Here's why. The molecules that are diffusing are either too large or they are polar. And we know that they cannot just simply slip through the plasma membrane. So let's draw out one of these examples. Okay, this is obviously a phospholipid bilayer, and I'm going to keep beating this to death until everybody knows the interior is nonpolar because of those lipids. So only nonpolar substances can pass through. However, you have this protein pump. So let's say I had these big old molecules. Doesn't matter what they are, but here's what you need to know. They're polar. Polar, 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 and polar. Well, there's a bunch of them over here. I'm going to draw a few more. Just draw them all over the place. And over on this side, there's hardly any. We'll just go in and write all this out just for the sake of it so no one gets confused. Polar, 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 and polar. So if we were in class, I'd ask you, which side is a higher concentration, the top or the bottom? It's definitely the top. And so these molecules want to diffuse through, and that is exactly what they're going to do. They're going to travel through this channel protein to get to the other side. That's their opening. This is facilitated diffusion. That channel protein is facilitating their diffusion from an area of high concentration to an area of low concentration. High, low. Got it? Okay. Now, for the third and final type of transport that is passive that you'll need to know, it is called osmosis. Remember, osmosis is passive transport, no energy required. Osmosis is the movement of water molecules across a membrane from an area of low solute concentration to high solute concentration. Now you might be thinking I just made a typo. Wait, what do you mean? Why are you going from low to high? That's not it. There's a lot here. This is the most confusing one, so I'm going to take my time and slow it down a little bit. In order to understand what's going on here, we need to first remember what a solute is. A solute is a substance that is dissolved in a solvent. A solvent is a substance that, the way I like to phrase it, it does the dissolving. So if you were to think of the Gulf of Mexico, the Gulf of Mexico has salt water. The solute is salt. The solvent is the water. When I was in school, like in junior in high school or sophomore in high school when I took bio, I never got that right. I could never figure out the two of them. So a little way to help you remember this is remember that water is nicknamed the universal solvent, and that might help you remember which one is which. Now, why does solvent have to do with this? Water is attracted to a solvent. If you want to do something really cruel and really mean, pour salt on a slug. And all the moisture that's in that slug is going to leave its tissues and go towards the salt, basically drying out the slug. Another example would be if you were to actually drink salt water from the Gulf of Mexico or from Tampa Bay or anywhere in the world. What happens is you drink the salt water, and the salt water has more salt in it than the salt that is in your own body, in your cells. And so the common rule here is, this is very important, water is attracted to the region with the most solute. So if you are drinking salt water, I can assure you that the salt water has more solute in it, more salt in this case, than the salt that is in your body. And so the liquid, the water that is in your cells and in your tissues is going to diffuse out of your cells and in your tissues and towards your digestive system because you just drank all that water. And that actually really dehydrates you. If you didn't know this, drinking salt water dehydrates you. Kind of a gross story. A few weeks ago, I took my dog to a beach park over in Pinellas County. And he's still kind of a puppy, so he hasn't been to the beach very much. And when he was in the water swimming around, he was drinking all that salt water. He was pooping straight up water for half the day, just salt water, no poop, no diarrhea, just salt water. Because all the salt that is in his cells and in his tissues was drawn towards the salt that he just drank. And so he had a lot of water in his digestive system, way more than he should have. And so he was just flushing out his whole system. And when we finally got him some fresh water, he drank a couple buckets full because he was so parched. Because all that salt basically dehydrated him. It attracted all the salt out of his, or all the water out of his cells. So let's make a drawing here to hopefully really make this give you that aha moment. And then I hope that you will understand it. And if it's challenging, don't be surprised. This is typically the hardest one to understand. So this purple line that you see here is going to be it's a membrane, but it's not going to be a phospholipid bilayer. This is a membrane in which only water may pass through. Okay? So let's put our water up in here. And we're going to put sodium ions all throughout this. Sodium ions are an atom of sodium that has one more proton than electron, so it has a positive one charge. So I'm just going to draw a whole bunch of these guys, okay? A lot of them. Lots of sodium. And over here, I'm just going to draw a few. Now, listen carefully. Here we go. According to this, as far as solute is concerned, the left is a high concentration, the right is a low concentration. But this is the diffusion of water. Osmosis is not the diffusion of ions. It is not the diffusion of sodium ions. It is the diffusion of water. So here is how you're able to determine which way water is going. You need to ask yourself, which side has more solute? It is clearly the left side. All this. Water is attracted to any area or any region that has more solute in it. So water is going to want to go in the left direction. Okay? That is one way to look at this. And when that happens, here's what you guys are going to find out. This area of water is going to rise, and this is going to lower until both sides are roughly equal. But if you look at it like this, you still see that there's more sodium on the left. So this way might not be the best to understand how this works. So here's a different way. And this is the one I would actually prefer for you. Let's get the water level back at being equal. You can come up with some arbitrary number. It doesn't matter what it is, but what you have here is salt water. So there's salt water here, and there's salt water here. What I want to know is what percent is solute and what percent is solvent? And we all know that percent set to equal 100. So you can make up some arbitrary number, but just make sure that it makes sense, okay? So let's just say that on the left, the amount of solute is 15%, and the amount of solvent, which is water, is 85%, okay? Now, over here, the solute. let's say it's about 5% solute, 95% solvent. I want you to ask yourself, which side has more water? The left or the right? We have 85%, we have 95%. I'm not a mathematician. I don't claim to be. I didn't really study a whole lot of math in school, but I do know that 95 is higher than 85. So when it comes to actual water, this is the high concentration, this is the low concentration. And according to the laws of passive diffusion and passive transport, which way do particles diffuse towards across the concentration gradient? Is it low to high or is it high to low? It is high to low. So the water is going to diffuse across until the difference between them is equalized. So as you add more water to the left, this side is going to become more diluted. And this side is going to become more concentrated. And so what's happening is the water level is actually going to change. So you can have water rising all the way up to here, and then the water on the other side is going to still be very lower. So you have less water. The amount of solute didn't change. This is going to be saltier water because it is a smaller volume. But this is going to be less saltier water because you added more of it. So if I were to try to give you another analogy here, imagine you have a bathtub filled with food coloring, okay? And it's like a bright green. What would happen to the color of that green if you turned on the faucet to the bathtub and allowed more water to fill the bathtub? Would the green get more brighter, which would mean it's more concentrated, or would it start to fade away, which means it's starting to dilute? The correct answer is it would start to dilute. And so you are decreasing the concentration of the solute in that bathtub. So by adding water to the left side here, we are decreasing the concentration of solute in the water. And by decreasing the water on the right side, we are increasing the concentration of the solute on the right side. I hope that makes sense. So now let's actually do some examples to begin to conclude, okay? So I'm going to make some regions here. Okay. So let's say in both of oops. There we go. Let me make sure you guys are still seeing what I'm seeing. I just have a little glitch there. Okay, good. We're all fine. Let's imagine that there's a semi-permeable membrane here, here, and here, okay? Now, stipulation. This, and this is a phospholipid bilayer. So you tell me, is that polar or is that nonpolar? It is nonpolar, so only nonpolar substances can pass through it. Let's get our water. Okay, so in this first example, I'm going to have oxygen molecules. We're going to put 5 of them. On the left, we will put 1 on the right. We're going to say that this is 0.5 molar, and we're going to say this is 0.1 molar, okay? Just stay tuned. Next, I'm going to have this box have hydrogen ions on the left side. Oh, I drew 1 too many. I just want to keep it at 5. And I have 1 on the left. So again, 0.5 molar, 0.1 molar. And then finally, over on this side, we're going to say I have sodium ions. 1, 2, 3, 4, 5. And then on the right, we have 1. And again, 0.5 molar, 0.1 molar. I'm going to actually give you some visual examples of simple diffusion, facilitated transport, and osmosis. All right? So let's draw some of these boxes again, and this is going to become like a before and after. Okay, this box is going to be simple diffusion. According to simple diffusion, do you need a channel protein to move across? No, you do not. Also, because I already stipulated this green line is a plasma membrane or a phospholipid bilayer, roughly the same thing. Polar and nonpolar substances can pass through. The answer is polar. And so what we have here is just 3 oxygens on the left and 3 on the right, because 2 of them diffuse through. So we have 0.3 molar and 0.3 molar. It's equal. Now, some people say that diffusion stops. It does not stop. What happens is diffusion continues, but it continues at an equal ratio. If 1 oxygen is going to diffuse from right to left, that means that 1 oxygen is going to diffuse from left to right. And this is going to be static. It's going to be, this is what we mean by it has reached an equilibrium, okay? The next one is going to be facilitated diffusion. We have our phospholipid bilayer. We have the water, and we have these hydrogen ions. But actually, before I start drawing them, if you already drew it, they'll be back. If you look up, hydrogen ions have a charge. And if I were a student in AP Bio and I wasn't in Chem yet, I'm gonna be like, how am I supposed to know if it's polar or not? I didn't take AP Bio, or I didn't take Chem. You see that thing right there? You see this symbol? That means it's positively charged. Does that mean that it has a charge? Yes. In chemistry, what does it mean to be polar? You have either a positive charge or you have the polar opposite, which means you have a negative charge. This is going to be polar. So can a polar substance like this molecule diffuse through this plasma membrane? The answer is no, negative. It cannot. So because I told you this is going to be facilitated transport, what you need are a couple channels. There's one. There's the other. So the hydrogen ions can pass through. So we have 3 hydrogens on the left. We have 3 hydrogens on the right. It has now reached an equilibrium, 0.3 molar, 0.3 molar. They pass through those channels. These gaps here are going to be channels, protein channels. Okay? Now, for the last one, the one that's the trickiest. Let's go ahead and get it all set up. There's a membrane. Here's our water. And I originally had sodium ions on one side and the other. Or I had 5 on the left and I had 1 on the right. So remember what I did over there. Which side, and think before you speak, which side has a higher concentration of water compared to the other side? Well, again, if you just make up some arbitrary numbers, you can do the same numbers I did for the example. Let's say that this side is 15% solute, 85% solvent, which means 85% water. Let's say this side is 5% solute, 95% solvent. That means that water is going to diffuse from left to right. Sodium ions cannot pass through that membrane because it is polar. But if you really know your biology, some of you might your ears might perk up. You say, wait, water's polar too. Water can't pass through that plasma membrane, and you're absolutely right. So what we have to do is we have to draw some pumps. These are going to be called, if you remember from lecture, these are called aquaporins. Aquaporins are a type of protein pump which allows water to diffuse across them, so it doesn't have to worry about the membrane. So with that being said, the water level is going to change. Water is going to diffuse from left to right. Or I totally said that wrong. I apologize. Right to left. It's going to diffuse from right to left. And that's going to cause the water level to actually change. Because we're diffusing from right to left, this side's going to be really high. This side's going to be really low. And so we have 1, 2, 3, 4, 5. And over here we have 1. Now it's going to still be equal. Remember, we're talking about concentrations and dilutions and all that. So this is still good. This could be 0.3 molar, and this could be 0.3 molar. You're like, well, I don't get it. This side is going to be the diluted side, so there's more water added, and this side is going to be the concentrated side, which means there's less water. Water was taken away. All right. There's one last thing I want to go over, and then we'll call it a day. And that's going to be the concept of hypertonic, hypotonic, and isotonic. So let's do this. Hypertonic, hypotonic, and isotonic. Hypotonic is a lower solute concentration. That's what hypo means. I messed that up. Darn it. Hyper means a higher solute concentration. Hypo means a lower solute concentration. And an isotonic means the same solute concentration. Okie dokie. So hyper means higher. Hypo means lower. And iso means the same. So let's actually put this to an actual test. I'm going to draw a beaker here, and we're going to do before and after. So here's our beaker. Inside the beaker, we're going to have some sort of solution, and we're going to have a cell, like a red blood cell. Okay? Now, let's say that inside the red blood cell is 0.4 molar sodium chloride, just some solute there. And you also have sodium chloride in the water, but the water is going to be 0.1 molar sodium chloride. Which region has more solute? Inside the cell or in the solution? The solution is what the cell is floating inside of. All this is the solution. What has more solute? If you said the cell has more, you are correct. And so, which way is water going to diffuse towards? Water is going to diffuse from the solution into the cell. And this is going to possibly cause the swell to swell up and maybe even burst. This phenomenon is known as a hypertonic solution. And I am tripping. It must be late because I think it is late. It's like 12:02 when I'm recording this. It is hypotonic. I had a student a long time ago come up with a little rhyme. Hypo like a hippo. So hypotonic, you need to really pay attention to the actual prefix here, guys, because you need to make it make sense. When we say that the solution is hypotonic, you have to understand that we're talking about the solution. You can look at this in two different ways. You can say the following. I can say that the solution is hypotonic to the cell, or I can say the cell is hypertonic to the solution. So you can say it's a hypertonic cell or it's a hypotonic solution. But the cell is going to swell up and possibly burst. Example number 2. Let's get back to having our cell. Let's say this is 0.4 molar sodium chloride. And out here it is 0.9 molar sodium chloride. Which side has more solute? Obviously the solution does. And so water is going to diffuse out of the cell towards the solution. What effect does this have? Well, water is going to cause the water level to rise, and that is also going to cause the cell to shrivel up. This is known as being a hypertonic solution. And as I said, this is subjective. You have to know which one you're looking at. If I'm looking at the solution, I would say the solution is hypertonic to the cell. Or we could say the cell is hypotonic to the solution. There's two different ways to look at it. Are you looking at this from the perspective of the solution, or are you looking at this from the perspective of the cell? Okay, and then the last example. We have our cell. Oop, wrong color. We have our cell, and inside the cell is 0.4 molar sodium chloride, and in the solution is 0.4 molar sodium chloride. This would be equal. Now, diffusion doesn't necessarily stop, but what does happen is for any water that diffuses out, you have the same ratio of water diffusing in. Same ratio diffusing in, same ratio of water diffusing out. And so the cell really doesn't change. It's still going to be the same. This is known as being isotonic. And it's going to be the same either way, but for just the sake of writing it, I'll write it out. The solution is isotonic to the cell, or you could say the cell is isotonic to the solution. Okay, that is pretty much it. That is the conclusion of passive transport. We will do another lesson which will cover active transport, which is the use of energy to go against the concentration gradient from low to high, and that requires the use of ATP, which is cellular energy. I'll see you then.